Good day, everyone. I will be discussing part two of my complement lecture. So, for this lecture, I'll be dwelling more on the alternative pathway and MBL pathway, which are very actually short topics. And if we still have time, I will discuss to you the roles of the cell-bound complement protein molecules, which serve as regulatory proteins. No, all right. So let's start with alternative pathway of the complement system. So basically, if you can still recall from my previous lecture, the alternative pathway is basically a complement activation pathway in which it is activated only when there is exposure to cell surface constituents. These cell surface constituents are unique to foreign cells or to invading organism. And we, unlike your classical pathway, alternative pathway does not need any antibody. In other words, the pathogens can be destroyed in the absence of an antibody. Thus, this pathway is considered antibody independent. Now, take note that the alternative pathway uses different sets of complement proteins. So remember, your classical pathway uses 9 complement proteins. Now we have a different names of proteins involved in your alternative pathway. For example, your C1, your C4, and C2 are not present, are absent in this pathway. No, these three now were actually present in your classical pathway but not in alternative. However, these are replaced by the following. So we have 4 serum proteins which are mainly involved in this pathway. So we have C3, factor B, factor D, and properdin. So remember in the nomenclature discussion, the proteins involved in alternative pathway are mainly termed factor followed by a capital letter. So in this case, we have two, no? Factor B and factor D. The properdin here is just actually stabilizing complement protein, which does not play a role in the activation. But still, no, it is part of the alternative pathway, but not in the activation of the cascade. All right, let's continue. So this was first described by Pellimer and colleagues in the early 1950s. So before, this was not actually termed as alternative pathway. This was called properdin pathway by Pellimer. But now, because they thought it was actually properdin which initiated the cascade, but now they discovered in the early 1950s that properdin is just a stabilizing agent. No, it just stabilizes the C3 convertase formed in the alternative pathway and does not cause activation. I mentioned this. Does not cause activation of your alternative pathway. So why do they call these alternative? Why did they call this alternative? It's because this is another way, an alternative way of activating your complement system. So this table shows you the different activators of your alternative pathway. So I showed a more or less similar table in classical pathway. So it's quite different in your alternative pathway. It's because if you can observe, this table actually enumerates the different components of that specific bacteria. Unlike your classical pathway, what's written there are the names of the bacteria which causes activation, direct activation of your classical pathway. Whereas in here, we actually have like lipopolysaccharides, no, the components components of the surface of the bacteria. Now, I want to direct your attention to this lower portion of the table na non-pathogen. So, you might be confused. Doc, you mentioned, Doc, that alternative pathway is antibody independent. How come now in this table it says here human IgG, IgA, and IgE. So again, these are actually exception to the rule. Why? It's because there is a prerequisite for this IgG, IgA, and IgE to activate your alternative pathway. That is, it has to be in complexes. Huh? Unlike your classical pathway, the IgG there involved and IgM actually are standalone antibodies. After that, they bind to the surface of the cell, and thus the pathway goes on. But for your IgG, IgA, and IgE to cause activation of your alternative pathway, it must be in complexes. Remember, IgA and IgE do not cause activation of your classical pathway. So these are other list of triggering substances for the alternative pathway. We have bacterial cell wall components. No, I mentioned this, lipopolysaccharide, capsules, teichoic acids. We also have cell walls, viruses, parasites, tumor cell lines, insulin, and dextran. So all of these serve as binding sites for C3BBB, one of the end products of this pathway. So do not confuse yourself. The C3BBB here is actually the C3 convertase in your alternative pathway. Later, I will explain this in detail. But for now, please take it in, no, by heart. The C3BBB is the C3 convertase in the alternative pathway. So, I will discuss that in detail. Do not worry. So, the first step in the alternative pathway starts with C3. Okay. So remember, comparing it with your classical pathway has this recognition, no? Recognition unit or recognition phase or stage 1. We do not have that in your alternative pathway because we start right away with your C3. What happens in C3, Doc? Remember, you mentioned, Doc, that C3 is only activated in the presence of your C3 convertase. How come now in alternative pathway, C3 becomes activated, becomes converted even in the absence of your C3 convertase? That's a very good question to answer. Now, to address that concern, what happens is that, remember, your C3 is the most abundant complement protein in the serum or in the circulation. And this C3 molecule actually circulates as an zymogen, as an inactive enzyme. So in the circulation, normally C3 is a zymogen. But when there are microorganisms that triggers the alternative pathway or microorganism substances, for example, the lipopolysaccharide, the teichoic acid, the capsule, and so on, this causes now your C3 to be hydrolyzed by water in the circulation. The water causes exposure of a thioester bond. The water causes hydrolysis of the thioester bond found in C3, causing C3 now to be proteolyzed or catalyzed into fragments, namely C3A and C3B. Therefore, without any antibody-antigen immune complex formation, C3, once there is an antigenic substance exposure, what happens is that C3 becomes hydrolyzed by water. And then your C3 becomes cleaved into two fragments, your C3A and C3B, for which your C3A is a smaller fragment, your C3B is a larger fragment. So which of these fragments participate in the cascade? The same with your classical pathway, the larger fragment participates in the cascade, which in this case is C3B. Do you get that? Okay, so we have C3B. Now, your C3B components actually binds to your foreign cells or invading bacteria or to the antigenic sites. So, Doc, you mentioned that C3B actually is the most abundant. Remember, being the most abundant, we have so many C3 in the circulation. Therefore, during the hydrolysis of C3 into C3A and C3B, we do not just produce one of each of these molecules. We actually produce many because we have more. We have abundance of C3 in the circulation. So not only one molecule of C3 is cleaved, but hundreds and thousands of C3 molecules. And so we produce many C3A and C3B. Now, the question is, Doc, how come now our own cells do not get affected with the uh, uh, activation of the alternative pathway? It's because of these reasons. Our cells, our own cells, our host cells, are actually rich in sialic acid. This sialic acid will actually hinder activity of your C3B, causing it to become inactivated. So supposing you have many C3B molecules that were produced by hydrolysis of your C3, what happens is that some of these molecules will actually be bound to our own cells. But our own cells do not get lysed. Why? It's because we have this protective mechanism. Our cells have high levels of sialic acid. And this sialic acid causes inactivation of your C3B molecule. Question, will presence of sialic acid causing inactivation of your C3B, will it cause the cascade to be further activated? Answer is no. Because once your C3B is inactivated, the cascade being a highly sequential manner, it will no longer proceed. So I hope you get that. And another reason is that once C3B is bound to our own cells, it becomes inactivated rapidly. No, that is due to the sialic acid, and I will discuss this later in detail, and the different cell-bound or cell surface complement proteins which are found in our own cells, absent in the cells of the bacteria or virus. So we have now a lot of C3B molecules. So we have C3B molecules bound now to the surface of the bacteria, virus, or any foreign cells. Now, in the presence of magnesium ions, C3B becomes bound to another complement protein participating in your alternative pathway. That protein now is called factor B. Again, in the presence of magnesium ions, C3B binds to factor B. Once your C3B binds to factor B, it actually produces another enzymatic complex. Another complex with an enzymatic activity. And what do we call that complex? I will discuss that in the next few slides. But just remember for now, alternative pathway starts with hydrolysis of C3. Hydrolysis of C3 produced via the thioester bond produces C3A and C3B molecules. Your C3b molecules are the molecules which participate in the cascade being the larger fragment. These larger fragment, the C3b molecules, bind itself to the surface of the parent cells, and in the presence of magnesium ions, it becomes complex to another form of protein in the alternative pathway termed as factor B. So C3b and factor B binds together in the presence of magnesium, forming a complex. What is the name of that complex? I will tell you later. But for now, let's discuss first the different components of your alternative pathway. We have factor B. So factor B has a molecular weight of 93,000 and is fairly abundant in the serum. Now take note that C3b must be bound to factor B for the alternative pathway cascade to proceed. Otherwise, if factor B is not bound to C3b, there will be no alternative pathway cascade that will happen. So again, it has to be bound to your C3b. You might ask, why Doc? How come factor B should be bound to your C3b? It's because once your factor B is bound to your C3b, C3b induces conformational changes to your factor B, making it more susceptible to cleavage or activation by another complement protein in the alternative pathway, which is termed as, very good, factor D. Again, I'll have to repeat that. Your factor B binds to your C3b. The main reason is that your factor B becomes activated only when it is complex or bound to your C3b. Now, once your C3b is bound to your factor B, it causes conformational changes in factor B, making it more susceptible to cleavage by factor D. Only bound factor B can be cleaved or cleaved by your factor D. So again, take note, C3b plus factor B forms C3b factor B complex. But with addition of your factor D, what happens is that your factor B becomes susceptible to cleavage. And so, cleavage of your factor B will actually produce two proteins, BB and BA. No more factor. No? You only use the word factor denoting that this complement protein is inactive. But once factor D binds to the complex formed by C3b and factor B called C3b factor B complex, this causes cleavage of your factor B producing BB and BA. So what is that complex produced? So factor D, its only substrate is bound. Take note, bound factor B. If you answer in the exam factor B only, I will mark it wrong. It has to be bound factor B because your factor B undergoes conformational changes when bound to your C3b. So in the presence of your magnesium ions, factor B is cleaved by factor D into two fragments. And what are these fragments? We have BA fragment and BB fragment. BA has a molecular weight of 33,000, whereas BB has a molecular weight of 60,000. So which of these complement proteins now is the larger fragment? Very good, BB. Which is the smaller fragment? BA. So which participates in the cascade? The larger fragment, which is the BB fragment. Therefore, upon binding of your properdin to C3b factor B complex, we now form, cleavage of factor B, we now form another complex we term as C3b, capital B, small letter b, or C3BBB, also known as your C3 convertase of your alternative pathway. What's the C3 convertase in the classical pathway? C4B2A. What's the C3 convertase of your Alternative pathway, C3b, BB. Classical pathway, C4b, 2A. Alternative pathway, C3b, BB. Classical pathway, C4b, 2A. Alternative pathway, C3b, BB. So your C3b, its only role is just to stabilize the alternative pathway C3 convertase. So listen here. So if we have a cell, and then this causes now an activation of your immune system, what happens is that your C3 becomes hydrolyzed. So hydrolysis of C3 produces how many fragments? Two, C3A and C3B. Your C3A is a smaller fragment which is an anaphylatoxin. Your anaphylatoxin binds the surface of your mast cells and basophils, causing degranulation, producing inflammatory substances, and thus releasing histamine. Whereas your C3B, being the larger fragment, participates in the cascade. Take note of that. So we have now C3B. What happens is that your C3B in the presence of magnesium ions has to be bound to what? Factor B. Factor B bound to C3B forms a complex named as C3B factor B complex. C3B factor B complex, spell out. C3B factor B complex. Now that complex is bound now, not really bound, no, but this binding of factor B to your C3B induces conformational change and thus causes factor B to be more susceptible to cleavage by factor D. And factor D, once it causes cleavage of your factor B, it produces two fragments of factor B named as BB and BA fragments. BB is the larger fragment, so this is the fragment that participates in the cascade. So you form now another complex C3BBB, which is the C3 convertase. So your C3 convertase, again, what's the target? Target complement molecule C3. No, from the name itself, C3 convertase. So it targets your C3 molecule. And your C3, take note, in the alternative pathway is very unstable, no, because C3BBB, no, it uses another, no, unlike your C4B2A, very stable, your C3BBB in alternative pathway is unstable. And so we need another complement protein named properdin, which causes stabilization of your C3 convertase. Now, addition of your properdin to the C3 convertase forms another complex we term as C3BBP. P for properdin. Again, C3BBP. So we call it stabilized C3 convertase. So do not worry, I'll show pictures later. So your stabilized C3 convertase cleaves more, and more C3, producing 3A and 3B. Some of these 3B now attaches to the stabilized C3 convertase, producing C3BBBPBB. Okay, do not get yourself confused. I will explain it in more detail in the next slides. Here, what happens in the circulation if you have an antigen that was recognized initially by your immune system or through immune surveillance? This causes your C3 molecules to be hydrolyzed. And once your C3 molecules undergoes hydrolysis via thioester bonds, it causes C3 now to be fragmented into two fragments, namely C3A and C3B. Now say B, activated. B, hydrolyzed. Say hydrolyze. Hydrolyze. Your C3 molecules now are hydrolyzed, producing C3A and C3B. Which of these molecules now participate in the cascade? 3A or 3B? Very good. 3B being the larger molecule, while 3A is a byproduct, no? an anaphylatoxin, which binds the surface of mast cells and basophils, producing immune inflammatory mediators and histamine. So we have a lot now of your C3b components. Now your C3b components bind to the surface of your antigen, of your bacterial cells. It has to be bound. Now, what is the next complement protein that gets activated after C3b formation? Very good, factor B. Now say factor B. So we have now factor B. And there is a prerequisite for factor B for it to become activated. What is that prerequisite? It has to be bound to your C3b molecule in the presence of magnesium ions. Say bind. 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 Your factor B now is bound to your C3b, forming C3b factor B complex. Is that active? No. Why? Factor B bound to C3b must be cleaved by another protein for it to become activated. And what is that complement protein? Very good, class. Your factor D. Factor D. Now, what happens is that in the presence of magnesium ions, your C3b now becomes bound to your factor B. And once factor B is bound to your C3b, what happens is that it induces conformational changes into your factor B molecule. And so your factor B molecule becomes an easy target for your factor D. And so when factor D causes cleavage now of your factor B, it produces two other fragments. Say cleave. Cleave. Your factor B now is cleaved into BA and BB fragment. Which of these fragments now participate in the cascade? Very good for your answer, class. BB, being the larger fragment which has a molecular weight of 60,000 kilodaltons. So you form now a complex called C3b-BB. C3b-BB. This complex is also known as the C3 convertase of the alternative pathway. And so, what is the complement protein that it targets? Of course, from the name itself, C3. So it can actually cause cleavage of so many C3, but there is a but. The C3 convertase formed by C3b-BB fragment is actually very unstable, such that it could not cleave all C3 found in the serum protein. And so it has to be stabilized by another complement protein for it to continue cleaving more and more and more C3 molecules. And what is that complement protein now? Very good, properdin. Can you say properdin? Yay. That is properdin. Now, properdin binds itself to C3b-BB, forming another complex we call C3b-BB-P, which is also termed as stabilized C3 convertase. Again, do not be confused, class. C3b-BB is termed as C3 convertase. C3b-BBP is termed as stabilized C3 convertase. Again, I have to emphasize again and again that stabilized C3 convertase versus C3 convertase are actually two different molecules because we added properdin into stabilized C3 convertase, which can now cleave more, and more C3 molecules. So again, the role of your properdin here is to cause stabilization of your C3 convertase such that it increases its half-life. So it could cleave more C3 molecules. Right. So it has to be C3b-BBP. P, there must be P after the BB. Now your C3BBB targets what complement protein? C3 convertase, because it is still C3 convertase, but it's just that it is stabilized, no, compared to the previous one. So it targets more C3. So more C3 are cleaved. Say cleaved, 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 and cleaved. Many C3b molecules now are cleaved, and this C3b molecules binds itself to the surface of the bacterial wall. And so this causes activation of more C3, 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 and this C3 sooner or later will be paired with another factor B. So again, as I have been telling you before, it is not only one molecule of your C3b that is cleaved. We need more for lysis to occur at different sites. No? Now, sometimes your C3b molecule that was cleaved by your stabilized C3 convertase may bind to the stabilized C3 convertase. So instead of binding to the surface of the molecule, it binds to your C3BBP, forming another molecule called C3BBP3B. This is considered the C5 convertase of your alternative pathway. See? C3B, BB, add letter P there, 3B. C3BBB, P3B is the C5 convertase of your alternative pathway. And by the name itself, this targets what complement protein? Your, very good, your C5 protein. C5 complement protein. Once your C5 now is targeted by your C5 alternative pathway C5 convertase, this causes cleavage of your C5, forming, so the usual cascade follows, no? The same cascade now, starting from C5. So C5 becomes activated, it is cleaved, say cleaved, cleaved. C5 now is cleaved into C5A and C5B. Your C5A is an anaphylatoxin, the smaller fragment which does not play part in the cascade, whereas your C5B now is the complement protein which plays part in the cascade. So, continuing the cascade, your C5B activates your C6, C7, C8, and C9, the same now. The same na siya sa classical pathway, forming eventually your MAC complex causing lysis of the cell. The same na yun. It only differs from activation up until C5 convertase formation. After that, the same na yun ang MAC complex formation. Okay, so I hope you got that easier, no, compared to classical pathway. Now let's talk about your lectin pathway. So your lectin pathway is also known as your MBL pathway or your mannose binding lectin pathway. So why is it called MBL or mannose binding lectin pathway? It is because this complement protein actually binds mannose binding lectin. So it binds to the mannose found in the surface of microorganisms. So your mannose is actually a carbohydrate on the surface of your microorganism. So what happens is that your mannose becomes bound to your MBL. MBL has components, namely MASP1, MASP, or membrane-associated serine protease, MASP1, MASP2, and MASP3. MASP1, MASP2, please add there, MASP3. Your MASP1 and MASP2 are structurally similar to your C1R and your C1S. So what happens is that your MBL your MBL actually binds itself, no, particularly your MASP1 and MASP2 binds to the mannose. Mannose. Your MASP1 binds to mannose. Now, and MASP2 binds to mannose. Once MASP1 and MASP2 are bound to mannose, what basically happens is that it causes now activation of your C4 and your C2. And then after that, the classical cascade follows. So the same cascade, no, easier, very easy. It just differs in the recognition or the activation, in which we need MASP1 and MASP2 to bind to MBL. MBL is found in the surface of the microorganisms or mannose binding lectin. So once your MASP2 and MASP1 binds to MBL, what happens is that it causes cleavage of C4 and C2. Cleavage of C4 and C2 produces C4b, C2a, C4b2a, which is the C3 convertase, and so on. The classical pathway activation cascade ensues. So I hope you're getting that. So this was the question I had thrown to you earlier, no? How come now your body or your own cells does not become bound to MBL? So the answer is that our own cells are rich in sialic acid, which are covering the sugar groups recognized by the MBL and are not a target for binding. So I have answered that already, no? Comprised of sialic acid. So let's proceed to our last topic for today. This is very short, very short. Control of your complement system. So how do we control the activation of your complement system? Now you might ask, Doc, in the first place, you were actually engrossed, you were very animated in discussing with the activation of the 3 different pathways. How come now we need to control the activation? Wherein, in the first place, we wanted the bacteria to die. Oh, were you thinking that way? Have you thought of that? Okay. Now, basically what happens is that when there is uncontrolled activation of your complement system, what happens is that it causes damage to your own tissues. No? Your own tissues. Remember, what are those byproducts in your complement system? We have your C2b, which is a vasodilator, increasing vascular permeability, leading to edema. Another, we have C3A, C3A, C4A, and C5A, which are anaphylotoxins. These anaphylotoxins actually induces release of your histamine and inflammatory mediators, causing now damage to your tissue. And so that's why we need system regulators. We need regulators to inhibit, to control the activation of the complement. So for example, once the target bacterial cell is dead now, you have to stop. And that inhibition, that inactivation is the function now of your system regulators. And again, who are these system regulators? These are actually I mentioned this. These are the cell-bound complement proteins or the cell surface complement proteins. And majority of these system regulators are aimed at halting accumulation of C3BY because C3 is the largest, the most abundant complement protein. So what are these control mechanisms? So the control of these mechanisms are directed at several levels. So remember, this is a cascade. And for us to stop activation, we must target the cascade itself, the different components of the cascade. So either we can regulate via the classical pathway, we can inactivate or target the C3 convertase being the major amplification process, no? I mentioned this. And we can also target the anaphylatoxins because anaphylatoxins are actually um, uh, byproducts which causes tissue damage. Okay, and of course we could stop or inhibit the MAP. So again, we regulate the classical pathway, we regulate the C3 convertase, we inactivate the anaphylatoxins and regulation of MAP. Class, it will be like this. I will not really ask you one by one, or I will not really be I will not vex you on this during the examination. No, I will not ask you the specific molecular weight, etc., etc. But what I will be asking are what I will be asking are the specific components found on these regulation sites. No, what are the components of the classical pathway that are regulated, the CT convertase, or those directed to anaphylatoxins and regulation of MAP? My exam will be like that. So for example, so there must be balance, no? For example, those substances which regulate the classical and lectin pathways. So we have number one, C1 inhibitor or C1 inhibitor glycoprotein. So why? Why does this inhibit the classical and lectin pathway? Remember, your C1 complex is the protein responsible for the activation of the recognition unit. Or in other words, C1 complex itself is the only protein making up the recognition unit of your classical pathway. And once your recognition unit is activated, the classical pathway cascade ensues. And so, deactivation or inhibition of C1 stops your classical pathway activation. Doc, how about for lectin? Diba lectin has no C1? Only MASP1 and MASP2. Your C1 inhibitor or C1-INH glycoprotein also inactivates MASP2. Thus, if MASP2 is inactivated, this now causes inhibition of your lectin pathway. So the cascade will not ensue. Another protein is C4b binding protein. C4b binding protein. So how come now, Doc? How come it inactivates your classical and lectin pathway? Remember, your C4b is the first protein to be cleaved by your C1 complex in the classical pathway. Once your C4 is cleaved by your C1 complex, particularly C1s subunit, what happens? It produces C4a and C4b. C4b is the complement protein which participates in the cascade, eventually forming with C2a, C3 convertase. And so if C4b now binds to C4, if C4b BP or C4b binding protein binds to C4, there will be no available C4b to bind to C2a that will form now C3 convertase. You get my point? So in other words, your C4BP will compete with your C2A. Will compete with your C2A for C4B. Mag-ilog ni sila, no? Ni C4B. And once your C4B becomes bound to your C4BP, what happens? It could not participate anymore in the cascade. And so C2A will be left alone. Oh. How about in the lectin pathway? Remember, once your MASP1 and MASP2 are activated, it causes activation of your C4 and C2. And so, once C4 is activated, it produces C4A and C4B. And when C4B binds to C4BP, there will be no available C4 for binding of C2A. No, same with the classical, no, same as a lectin. Next, we have complement receptor type 1, which is also known as CD35. Complement receptor type 1, which is also known as CD35. Again, I will not vex, no? I will not vex you that much. I will not let you memorize all of these. No, even me, I was not I have been teaching this topic for 4 years already, and still now I am not able to memorize this bullet here. No, so the more that I will not let you memorize, what I want you to know is that CR1 or complement receptor type 1 is also known as CD35, and that your CD35, it targets your classical and lectin pathway. Classical and lectin pathway. So it inhibits formation. Take note also, it inhibits formation of your C3 convertase in the classical and lectin pathway. So it targets the classical and lectin pathway. What particular component? The C3 convertase. So this also binds to C3b and C4b but has the greatest affinity for C3b. And this also mediates transport of C3b to the liver and spleen, no? Also termed as immune adherence. Now, another protein is what we call MCP or membrane cofactor protein. You need to memorize its other name, the CD designation, which is CD36. Again, CD46 rather. CD35 is also known as the previous one, okay? While CD46 is also known as, all right, okay. So, this inhibits formation of C3 convertase in the classical and lectin pathway. Next, we have decay accelerating factor, also known as CD55. CD35 is also known as, you answer that. CD46 is also known as, CD55 is also known as this one. So this inhibits again C3 convertase in the classical and lectin pathway. This also is capable of dissociating both the classical and alternative. Take note, it has also action in the alternative pathway C3 convertases. So it targets all the three. Next, we have regulation of the alternative pathway. What are these proteins? So we have factor H, which acts by binding to C3b, thus preventing the binding of factor B. So it binds to C3b so that factor B now could not bind to C3b. So what complement protein does it prevent from forming? Your C3 convertase. Because remember, your C3b BB is the C3 convertase. And once your factor B is not bound to C3b, what happens? There will be no activation of the cascade. So it also acts as a cofactor that allows factor 1 to break down into C3b. Next, we have S protein, which is also known as vitronectin. So it binds to C8 and C9, but it interacts with the MAC, no? The terminal components or the MAC complex. It binds with C5b, C6, C7, C8, and C9. But there is no polymerization that occurs when S protein binds to C9 also. Next, we have the Merl complex, also known as CD59. Again, CD35 is known as CD46, CD55, and then how about CD59? Okay, so you memorize that, huh? So this blocks formation of the membrane attack complex by binding to C8 and prevent insertion of C9 into host cell membranes. And then lastly, we have homologous restriction factor, which prevents formation of the MAP on the surface of autologous normal cells. So this part here will not come out in the examination, but let's just run through this. No? So remember, I mentioned you that once your C is followed by R, that means that is a receptor. That protein is a receptor. And the function of that protein is to actually where your complement proteins bind to. So your CR1 or receptor type 1 is the receptor for C3b and IC3b, whereas your CR2 or receptor type 2 is the receptor for IC3b, and these are actually found in B cells. And then we have receptor type 3 or CR3 a receptor for your IC3B. Class, do not memorize. These two slides will not come out in my exam. Next, we have C4, which is a receptor for IC3B. So this will not come out in the exam also. But I have to run through that for board exam purposes. Next, we have anaphylatoxin receptors, C3A, C4AR. You just add R. The R there denotes that is a receptor. And we also have your C5A receptor. These anaphylatoxin receptors are found in your mast cells and basophils. And upon binding of your complement protein C3A to C5A, this causes degranulation of your mast cells and basophils, releasing inflammatory molecules and histamine. So these are the this is just a review of what I have mentioned in the previous slides. So again, these are the byproducts, byproducts of your complement activation. So again, what are the anaphylatoxins? C3A, C4A, and C5A. What's the function? What's the role of your C3? Your anaphylatoxins? It causes release of histamine from basophils and your muscles, increasing vascular permeability. Next, we have chemotaxin. So your 5A, C5A, serves as a chemotaxin. So you learned this from your innate immune system lecture that your chemotaxin causes or induces chemotaxis. So your chemotaxis is when your neutrophils, your other phagocytes become invited or goes near to the source of your antigen for phagocytosis. So that is what we call chemotaxis. Plus, what we need to remember is that C5A is a chemotaxin aside from being an anaphylatoxin. Next, we have opsonization. So opsonin, what are the opsonins I've mentioned? Okay, the B components. So we have the C4B, C3B, and IC3B. These are all opsonins. And deficiency of your complement proteins, if you do not have complement proteins, this may actually make you susceptible to infections. Why infections? Because your complement protein actually causes destruction or lysis of your bacterial cell. So what I actually presented a table in your notes, on the different deficiencies associated with complement proteins. So you can read on that. I might get 1 to 3 questions from that table alone. So plus, I'm actually helping you pass the examination. So you have to do your part. I'm giving out pointers already. So last topic, last few slides, we have evaluation of the complement pathway. So this is our job as medtechs. So what do we do in the lab? So basically, remember there is what we call in the previous slide complement deficiency, meaning you are lacking some complement proteins in your serum such that you are now more prone to infection. So how do we know what particular complement component is deficient in your serum? Okay, so we have 2 ways here, or 2 basic steps. The first step is the overall determination of the function of the complement, and then the second step is to determine the particular component that is deficient. So again, first, in other words, we determine which complement pathway is deficient. So we test if is it the alternative, is it the classical pathway. Unfortunately, we do not have tests for the lectin pathway available in the lab. We only have classical and alternative pathway. Once we know already which complement pathway is deficient, now we can determine which of those proteins under that pathway is deficient or is lacking or is not present. So I hope you understand that. So let's discuss first the first step. So the first step Our first test is comprised of two tests. The tests are named as CH50 and AH50. C stands for classical, A stands for alternative. So which of these pathway tests for deficiencies in the classical pathway? CH50. How about in the alternative pathway? AH50. Now, if your CH50 is positive for deficiency, whereas your AH50 is negative for deficiency, meaning there's nothing deficient there, what we do is we test for individual proteins, complement proteins found in the classical pathway. On the other hand, if your CH50 is negative for deficiency, whereas your AH50 is positive for deficiency, we now proceed to step 2, that is to test the different complement proteins found in your alternative pathway. So I'll give you an example later. So how do we do CH50 and how do we do AH50? So just take note of the materials used here. So we have sheep red blood cells incubated with anti-sheep red blood cells, IgG antibodies, to form immune complexes, no? Antigen antibody immune complexes. Because why? CH50 is for classical pathway. And so we need formation of our antibody complexes. And the positive result is actually formation of your lysis. Formation of your lysis. When serum is added to the culture complexes, the sheep red blood cells are lysed if the complement is intact. No, that is the result that we want. But meaning if there is lysis, your complement proteins in the classical pathway are complete. We do not have deficiency. But if there is no lysis, remember, we need each of these components to cause lysis, or each of these components are activated sequentially. If one is absent, we could not produce lysis. So a negative result, a positive result for deficiency or presence of deficiency is actually noted by absence of lysis. Again, absence of deficiency is noted by presence of lysis, meaning no deficiency when there is lysis. Presence of deficiency would mean there is absence of lysis, meaning there are proteins which are not found in the serum of the patient. And next we have the AH50. So we use rabbit this time, erythrocytes incubated with the serum of the patient. Now, the same results that we get, no? In the presence of serum containing complement proteins, the rabbit erythrocytes are lysed in the presence of protein component. But if these serum complement proteins are absent, the erythrocytes do not lyse. Okay? And lastly, that is step 1. After step 1, we can now do step 2. So if any of these AH50 or CH50 is abnormal, we can now determine which specific complement protein is deficient with each pathway. So this can be done either 3 or 2 method, or could be 3, could be 2 methods from other books. So other books mention 2, other books mention 3, but you have to use my notes or use my PowerPoint as your reference for the examination. So we can use RID. So I will not discuss this in detail because you will have a separate lecture for this. What you need to remember are the names, no, of the method. So we have RID, and then we also have nephelometry. Okay, so that ends my lecture for the complement system. If you have questions, I am available online 24/7. Yes, you heard it right, 24/7. You can email me at Brian Denny at cim.edu.ph. All small letters. Brian Denny, no space, at cim.edu.ph. Okay, so let me end my lecture with this quote. I know you are third year guys, and I know you feel overwhelmed with many subjects no, that you have to study. But again, when you feel like quitting, think about why you started. Take note that you're here in third year for a reason. You're here in third year because you made it to second year and first year. You made it this far. So there's no reason for you to quit. Even if you fail in some of the exams, no, try to laban, try to bawi lang. Think about why you started. So let me end my lecture with a song number I have prepared so that this information will be instilled into your minds. You can sing with me if you like. Okay, so here's the song. It's titled Compliment. Hey guys, this is your song. Talk about complementivation. This is described as a cascade. There are three major complement pathways. What are these? Classical, alternative, and Manos. This was first described by Bordet, coined by Mr. Ehrlich, just gotta say. Activation is described as a cascade. Numerous proteins are sequentially activated. Products covalently attached to microbial surface, which promotes opsonization and salicysis. Start tweaking like C1, C2 and C3, C4 and C5. Yo, work it like math. The classical pathway needs antibody. It needs IgM or IgG. Alternative pathway and lectin pathway are antibody independent. Oh, it promotes opsonization and lysis of pharynx cells. This was first described by Bordet, coined by Mr. Ehrlich, just gotta say. Activation is described as a cascade. Numerous proteins are sequentially activated. Products covalently attached to microbial surface, which promotes opsonization and salicysis. Start looking like C1. So and C3. Join me guys. C4 and C5. Look it like mahap. Thank you everyone. Have a good day. Do not hesitate to email me if you have questions. Thank you very much.